Yeah. Shahin's score is 530 out of 720. That is really great, my dear. So keep it up and try to get more marks, okay? All right. Good, very good. Okay, now let's enter into the class. Good evening, very good evening to all of you students. So yesterday we have started the biomolecules lesson, right? So yesterday we have the learning objectives of this lesson are carbohydrates till hormones. Yesterday I have discussed about this. And yesterday we have completed the classification on the basis of behavior on hydrolysis. So there were three classification under this. Yesterday we have completed the monosaccharides and oligosaccharides, right? So in oligosaccharides, mainly we have discussed about the disaccharide part, right? We have discussed about the disaccharide part in yesterday's session. So who are the three disaccharides we have discussed? Who are the three disaccharides we have discussed? Can anyone tell me? The first one is sucrose. The second one is maltose. And the third disaccharide that we have discussed is lactose. Right? And in case of monosaccharide, we have discussed about glucose and fructose, right? So today we are going to see about the polysaccharides. So let's enter into the polysaccharides topic. Okay, so there are three polysaccharides which we are going to discuss. Okay, there are three polysaccharides. So yesterday itself we have studied the definition of polysaccharide. So who are polysaccharides? The compounds which on hydrolysis with dilute acids or enzymes produces more number of monosaccharide molecules are called as polysaccharide. Okay. So if you are hydrolyzing a polysaccharide, you will end up with more than 10 molecules of monosaccharides. Got it? So those are called as polysaccharides. So you just see this picture. What do you observe from this picture? Two monosaccharide units are coming, and on hydrolysis, they are forming the trisaccharide, and one more molecule will come, and that will come and attach with it, and it will form the another glycosidic linkage bond. And the fourth molecule can come, and the loss of water molecule will lead to the tetrasaccharide. So like this, the chain goes on. Okay. So they are called as polysaccharides, which has more than 10 molecules of monosaccharide. Got it? So they mainly act as a polysaccharides mainly act as a food storage or structural materials. The examples for polysaccharides are starch, cellulose, and glycogen. All right. So let us discuss one by one. Let us study the structure of each and every polysaccharide. Okay. So first let me go for the structure of starch. Basically, starch is a polymer of alpha glucose. Okay. It is a polymer. What is a polymer? First of all, more number of monomers will join together to form a polymer. So starch is a polymer of alpha glucose. This is very important. It is a polymer of alpha glucose. Now let's study the components of starch. There are two components. One is amylose and the other one is called as amylopectin. And I am going to discuss some very important points about amylose and amylopectin, which are very, very important. Okay. Amylose is a unbranched chain polymer of alpha glucose. Okay, it is an unbranched chain polymer of alpha glucose. And there will be the presence of carbon 1, carbon 4 glycosidic linkage in amylose. Okay, and it is a water soluble component which constitutes about 15 to 20% of the starch. Okay, only the minor portion of the starches are composed of amylose. Okay. In the other side, when you come to amylopectin, it is a branched chain polymer of alpha glucose. Okay, branched chain polymer. And here, if you look into the glycosidic linkage, we have two types of glycosidic link. One is between C1 and C4, and the other one is between C1 and C6. Okay, so two glycosidic linkages are present in amylopectin. And unlike amylopectin are insoluble in water, and they constitute about 80 to 85% of the starch. Okay, so the major portion of the starch is composed of amylopectin. Okay, 85%. And amylose is of just only 15%. Got it? 15 for amylose and 85 for amylopectin. Can you all able to get the points which I am discussing here? I will discuss the structure of amylose and amylopectin in the next slide. Don't worry, I will show you the structure also. I will tell how it is, why it is called as unbranched and why amylopectin is called as branched. Let us discuss the structure also. Any doubt in this for anyone? You just need to take down the points. That's it. Nothing much about the concepts. Okay. You just need to memorize few things from this two types. That's all. Shall we go for the next slide, students? Do you have any doubt from this? So each and every point is very important. So please have a note of it. Shall I move? Okay. Thank you so much, students. Now let me go for the structure of amylose. So it is an unbranched chain polymer, right? So listen. So it is a polymer of alpha glucose. And if you remove the water molecule, you will end up with the structure of amylose. So it is an unbranched, so there is no branch. So it is a linear polymer. It is a linear, it is going in a straight line, right? So it is a linear polymer. And you all know, we have the oxygen linkage is called as glycosidic linkage, and we have carbon number 1 and carbon number 4, C1, C4 linkage in amylose. Okay. So you don't have to memorize the structure. Okay. Memorizing is very easy only, but you don't have to worry much about it. Even if you forget, you can recall the structure of glucose. Yesterday we have discussed the structure of glucose, right? Alpha glucose and beta glucose. So if you remember the structure of glucose, you can get the structure of amylose, because we are repeating the unit of this glucose molecule and forming the amylose, right? Even if you forget, no issues. You can draw the structure like this separately, remove the water molecule and connect it with the glycosidic linkage. Clear? So here we are, the bond is shown like this. So this is called as the polymer. It means the chain goes on. Okay, the chain goes on in a linear fashion like this. So this is all about the structure of amylose. Then talking about amylopectin, it is a polymer, or it is also a polymer of alpha D glucose, but here we have both branched and a linear manner. So we have C1, C4 linkage. So here if you see, we have C1, C4 glycosidic linkage. And the branch, if you see, we have C1 and C6 glycosidic linkage. Carbon number 1 and carbon number 6 of the other chain is forming the glycosidic linkage here. Okay, so this is C1, C4. This one is C1, C4 alpha linkage. And this one is C1, C6 alpha linkage. It is present in amylopectin. All right. Can you understand? So this is all about the informations of the first polysaccharide, which is starch. Okay. So now let us go for the second polysaccharide, which is called as cellulose, which is a cellulose. Cellulose is a straight chain polymer of beta D glucose. Beta D glucose. Okay, starch is a alpha D glucose polymer and cellulose is a beta D glucose polymer. So take the beta D glucose structures. Okay. remove the water molecule and connect. So you will get the beta linkages. Okay, you will get the beta linkages. So this is called as C1C4 beta linkage. C1C4 beta linkage. All right. Clear? If you are taking an alpha monomer, you will end up with alpha linkage. If you are taking a beta molecule, you will end up with beta linkage. Okay. If both are on the same side, it is alpha. If they are in the zigzag fashion, it is beta. If you look into the first carbon or first molecule here, the oxygen is above. But for the second molecule, the oxygen is below. So they are like this. Okay, they are like this. So it is a beta linkage. So in cellulose, we have C1, C4 beta linkage. So these are the two important points you have to keep in mind regarding the structure of cellulose. Now let me go for the third polysaccharide, which is called as glycogen. What is a glycogen? Carbohydrates are stored in animal body as glycogen. Okay. So carbohydrates are stored in animal body as glycogen. So this is the body. Okay. And we have the brain here, right? We have the brain and these are the parts of our body. When the body needs glucose, okay, when the body needs glucose, the glycogen takes with the help of the enzyme and it breaks down as glucose. Okay. Glycogen is a kind of storage. So when your body needs glucose, the enzyme will break down the molecules of glycogen into glucose and the glucose will be useful, okay, for doing the actions and for doing some works. Clear? This is all about the glycogen. So with this, the polysaccharide topic is also getting over. Do you guys have any doubt from starch, cellulose and glycogen? Shall we go for the second biomolecule? So now only we have completed the first biomolecule, which is carbohydrates. Okay. So carbohydrates is a big topic of this lesson. So we are completing that here. Do you guys have any doubt in carbohydrates? Shall we enter into the protein? Are you all ready to listen the protein topic? Do you have anything to ask from carbohydrates? Monosaccharide, oligosaccharide, and polysaccharide. Under each topic, we have discussed many things. So just remember the flowchart which I have given you at the beginning. Okay, so if you remember that, everything will be easy. So there were some few points that you have to keep in mind or you need to memorize under each topic. Rest are fine only. We did not study any big reactions here. Only under the glucose topic, we have studied some reactions. Apart from that, there is no reaction. Okay. Students, tell me, shall we go for proteins? Am I audible? Why are you all staying so silent? Okay, thank you, Bhagat. Now let us go for protein. If you want to study about proteins, you need to know some information about amino acids. Okay. So let us study about the amino acids and their classification first, and then we will go for protein. Okay. So let me start with amino acid. What are amino acids? All proteins are the polymers of alpha amino acid. This is the very important point you should know. Proteins are the polymers of alpha amino acid. Okay. Starch is a polymer of alpha D glucose, right? So in the same way, proteins are the polymers of alpha amino acid. The simplest amino acid is called as students, am I audible? Hello? Can you able to hear my voice? Yeah. Okay. The simplest amino acid is given to you on the screen. Okay, and this is the formula for the simplest amino acid, and this is a glycine molecule. Okay, so we have carbon, and the carbon is attached with two hydrogens and one NH2 group and one COOH group. You all know that COOH is an acidic group and NH2 is a basic group, right? So we have different types of amino acid, alpha amino acid, beta amino acid, and gamma amino acids. So for example, if I take an amino acid like this is an alpha amino acid. So why it is called as alpha amino? Because the amine group is attached to the alpha carbon. What is an alpha carbon? The carbon which is adjacent to COOH group is called as alpha carbon. Okay. So that's why it is called as an alpha amino acid. So what about this amino acid? Tell me whether it is alpha, beta or gamma. Is it alpha or beta or gamma? So this one. The simplest amino acid is an alpha amino acid or beta amino acid or gamma amino acid. Very good. It is alpha amino because the amine group is attached to the alpha carbon which is adjacent to COOH. Okay. So this is an alpha. Now let us study the classification of amino acid. So it is classified on the basis of acidic and basic nature. And secondly, it is classified on the basis of source. Okay. So first let's go into the classification on the basis of acidic and basic nature. Got it? So according to this classification, amino acids are getting divided into three. Okay. First one is acidic. Second is on the basis of basic. Third is on the basis of neutral. So what are acidic amino acids? If the COOH group in the compound is greater than NH2 group, compound means the amino acid compound. Okay, if the amino acid has more COOH, then it is called as acidic. Okay, here you see in aspartic acid, we have two COOH group. This one COOH and this is another COOH. But we have only one NH2 group. If the carboxylic acid group is greater than NH2 group, it is called as acidic amino acid. Okay. And next, basic means NH2 will be greater than COOH. So in this compound, you see we have two amine group and only one COOH group. This is lysine molecule and this is a basic amino acid. And if both of them are equal, then it is called as neutral amino acid. The example is alanine. Okay. So here if you see, we have one NH2 group and one COOH. If we have two NH2 group, there will be two COOH. Okay. If the amino group and the acid groups are equal, then they are called as neutral amino acid. Clear? Very easy. So this is the first classification, which is on the basis of acidic and basic behavior. Now the second classification is on the basis of source, right? So according to the source available, it is classified into two. One is called as essential amino acid and the other one is called as non-essential amino acid. So what is meant by essential amino acid? So the one which is essential for health and to be supplied through diet. The particular compound is very essential for you. Okay, but it is not present inside your body. So what you have to do? You have to take those essential amino acid compounds through the food only. Right? So those are called as essential amino acids. And the non-essential amino acid means your body itself can synthesis. So it is non-essential amino acids are the compounds which can be synthesized by our body. It is not essential. Not essential means you don't have to take those amino acids from outside. It is not essential for us. Our body itself is capable of synthesizing those amino acids. So those are called as non-essential amino acids. Clear? Now let us see the essential amino acid types and non-essential amino acid types. there are 10 essential amino acid and 10 non-essential amino acid. As I have already told you, so this is the general formula for amino acid where we have one carbon and it is attached to 4 different groups. Okay. And this is a list of 10 essential amino acid. So the first column is the name, valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, tryptophan, and histidine. Okay. So these 10 are the essential amino acid and these are all the molecules. Okay. Formula for the molecules of these amino acids. These are the 3-letter symbol. 3-letter symbol for the amino acid. This is the 1-letter code. The first letter will be mostly taken as a code for that particular amino acid. Just need to memorize the names of the amino acid. Clear? That is enough. Now let me go for the non-essential amino acid. So these are the 10 non-essential: glycine, alanine, glutamic acid, aspartic acid, glutamine, asparagine, serine, cysteine, tyrosine, and proline. So these are the 10 non-essential amino acid. Okay. And you see here glycine. So the R group of glycine is hydrogen. The R group of the glycine is also hydrogen. So if you substitute hydrogen, then this carbon is not a chiral carbon, right? Because 2 groups are same. 2 groups are hydrogen. So it is optically inactive amino acid. The only optically inactive amino acid is glycine. The only optically inactive amino acid is glycine. I will discuss about this in the upcoming slides, okay, in a detailed manner. So you just keep in mind that the only optically inactive amino acid is glycine, whereas all other amino acids will have 4 different groups attached to the chiral carbon. Okay, and glycine is an example for non-essential amino acid. Clear? So students, can you all able to understand what I am teaching? Can you able to follow? Do you have any doubts? You don't have to worry about the formulas of amino acids here and the 3-letter symbol and 1-letter code and all. You just study the names of the amino acids. That is enough. Shall we go for the next slide? No questions have been asked from names also, but for the safer side, you can study those. Okay. Shall I go for the next slide? Any doubts so far? The next is about the properties of amino acid. Shall we go to that? Okay. Let's discuss the properties. First of all, amino acids are colorless crystalline solid. You can able to see it in the picture, right? There is no color, but it is crystalline in nature. Some needle kind of crystals are present, right? So it is a crystalline colorless solid. And they are water soluble. If you put inside the water, they will get dissolved and behave like a salt. Okay, like we are dissolving NaCl in water. The same way you can dissolve this inside the water and it will get dissolved. And amino acid forms Zwitter ion. So what is meant by Zwitter ion? What is meant by Zwitter ion? If your compound has both positive charge and negative charge, then that compound is called as a Zwitter ion. Okay. So let me take one amino acid. So here I have NH2 group and COOH group. So I can form the Zwitter ion from this molecule. How? The lone pair of nitrogen will go and attack the acidic hydrogen of carboxylic acid. Okay. The carboxylic acid hydrogen is acidic. So since the lone pairs are electron-rich center, so the lone pair electron-rich will go and attack the electron-deficient site. And the OH bond will be broken and the electrons will move towards oxygen. Okay. Now this molecule is in equilibrium with this molecule. So what happened? The hydrogen has moved to NH2 and the electrons have jumped to oxygen. So this compound is the zwitterionic form of the amino acid. Why? We have positive charge and negative charge. So that's why this compound is called as zwitterion. Okay. One more point you should know about this zwitterion is it is amphoteric in nature. What is meant by amphoteric? The one which can act as an acid as well as base. The one which can act as an acid as well as base is called as amphoteric. So zwitterion is amphoteric because it can act as an acid as well as a base since it has both positive and negative charge in it. Okay, clear? And also it is neutral. Zwitterion is neutral in nature. Plus and minus, one plus and one minus, right? So they'll get cancelled with each other and it is neutral. So these are the points you should know about the zwitterion and the amino acid. So this is the third point, right? This is point number 3. So amino acids are colorless crystalline solid. They are water soluble. They can able to form zwitterion. Zwitterion is neutral and amphoteric. Zwitterion means both positive and negative charge will be present. Okay. Shall I go for the fourth property? Do you have any doubt from this zwitterionic part? Can you all make the zwitterion if I give some other amino acid to you? Whatever might be the amino acid, you just have to take the lone pair of the amino group and attack the hydrogen and write the structure. That's all. Shall we go for the fourth property? Students, are you there? JNV Rowing, Madhuka Reddy, Jabrun Sultana, Sivamani. Am I audible to all of you? Shall we go for the fourth property? Okay. All right. So the fourth property is the optical properties of amino acid. It already taught, right? Optical activity. So if a compound is having chiral carbon, it is optically active. So what is meant by a chiral carbon? The carbon which is attached with 4 different atoms or groups is called as chiral carbon. So let me take this amino acid. This carbon is an asymmetric carbon. Asymmetric carbons are otherwise called as chiral carbon. Okay. This is otherwise called as chiral carbon. Carbon which is attached to 4 different atoms or groups is called as chiral or asymmetric carbon. Okay. So here we have hydrogen in one side and CH3 on the other side and NH2 is the third group and COOH is the fourth. So all 4 of them are different. So that's why the central carbon is a chiral carbon. Okay. So if a compound is having chiral center, then that compound will be optically active. This is the criteria. One of the criteria for a compound to be optically active. If a compound has a chiral center, then it is optically active. So amino acids are optically active except glycine. This is what I have discussed in the previous slide. Okay, this is what I have discussed in the previous slide. Got it? The only optically inactive amino acid is glycine. So this is a structure, right? So why glycine is optically inactive? Because the R group is hydrogen. So because of this, we are ending up with 2 same groups. Or only if all 4 groups are different, then it is chiral. If 2 of them are same, then it is not chiral. It is an achiral compound or it is not an asymmetric carbon. Okay, so that's why the glycine is optically inactive. Okay, optically inactive compound. A very important point. Now the fifth property of amino acid is about the D and L amino acid. So D amino acid means the amino group will be present on the right side, and L amino acid means it will be present on the left side. That's all. Okay. Yesterday we have discussed for glucose and fructose, right? If the last before carbon has OH on the right side, it is D, and if they are present on the left side, it is called as L. The same concept you have to apply here also. Got it? So these are the informations about the amino acid. So we have studied the classifications and the properties of amino acids. Now let me discuss about the peptide bond. Peptide bond in protein. Okay. Proteins are the polymers of alpha amino acid. So this is a statement I have discussed in the beginning of protein. Right. In the beginning of amino acid, I have given this statement. Proteins are the polymers of alpha amino acid. And now the amino acids are connected to each other by peptide bond or peptide linkage. How? So let me take two amino acid first of all. The first amino acid is glycine, and the second amino acid that I am going to take is alanine. I have taken two amino acid, and what you have to do, you have to remove the water molecule. You have to remove the water molecule from both the amino acid. Okay. OH from the acidic part of one molecule and hydrogen from the amine part of the other molecule. Got it? So this is how you have to remove the water molecule and connect. Connect CO and NH. Connect this C double bond O and this NH. Okay. So you will get a compound which is called as glycyl alanine. And this is the peptide linkage. Why it is called as peptide linkage? C double bond O NH, right? Actually, we call this linkage as amide linkage, right? This linkage is called as amide linkage. But since this linkage is present in proteins, we are calling this as peptide linkage. Okay, peptide linkage. So this is a polymer. Okay, this is a polymer. Or this is called as a protein chain. Got it? So this is how we form the protein chains by connecting two or three or more than three amino acids. Is it clear to all of you? If you want to make alanine glycine protein chain, okay. So first you have to draw the molecule of alanine, then you have to draw the molecule of glycine. Okay. So from alanine you have to remove the OH, and from glycine you have to remove the H. So you will end up with different protein chain in that case. Okay. So the placement of each amino acid is very important. Is it clear to all of you? Can you all form the peptide bond if I give some other amino acid pairs to you? Any doubt from peptide bond formation for anyone? Shall I go for the next topic? We have discussed about the amino acid and their classification and the peptide bond. Okay. So this information is enough to study about the protein. Now let's go for the protein and its classification. Okay. So now the protein is classified on the basis of their molecular shape, first of all. According to their molecular shape, it is classified into two. One is called as fibrous protein and the other one is called as globular protein. So in this biomolecules lesson, if you see, we are going through many types and classifications. Okay, so what I suggest you guys is, please write down all the types and all the classifications and all the divisions which I am giving you on the slide. Okay, write down everything in your notebook and study. Otherwise, you will get confused which one will come under which type. Got it? So now we are studying about protein classification. The first one is on the basis of molecular shape. According to it, it is getting classified into two, fibrous and globular. So fibrous means it is a fiber kind of thing. Okay, this is the picture. fiber kind of picture you will be having. And it is a polypeptide chain which is running parallelly. Okay, one chain is parallel to the other, right? Polypeptide chain run parallel here. And when you go for globular, it is like a globe. Globe means it is spherical kind of structure, right? So they will, all the polypeptide chain will coil around with each other and they will form a globe kind of structure. Okay. So fibrous protein is a fiber-like structure. Globular protein is a spherical shaped molecule. And fibrous proteins are insoluble in water. Globular protein is soluble in water. The very important point is the example which I am going to give. The examples for fibrous protein is keratin and myosin. So keratin is present in hair, okay, in our hair, in wool and silk. Myosin is present in muscles, okay. Each and everything is important. Keratin is present in hair, wool and silk, and myosin is present in muscle. And coming to the examples of globular protein, insulin and albumin are the examples of globular protein. Insulin and albumin. Clear? Can you guys able to understand the classification on the basis of molecular shape? Shall we go for the next one? Next classification. I have discussed all the points under fibrous and globular. Each one is important. So here the chain runs parallelly. It is running parallelly, right? And here they are coiling around and forming a globe kind of structures, the spherical shape. Spherical shape. They are coiling around and giving the spherical shape, and here it is fiber-like structure. Got it? Students, shall we go for the next? Shall we go for the next classification? Shall we classify based on the structure and shape? Yes or no? Only one student is saying yes. I don't know what others are doing. Are you all sleeping? Okay. So the second classification is on the basis of their structure and shape. On this basis, it is classified into 4. Primary structure, secondary structure. Secondary structure is having 2 classifications. One is alpha helix and beta pleated sheet. Third is tertiary structure and the fourth one is quaternary structure. So according to the structure and shape, it is getting classified into 4. Okay. Now let us discuss one by one. So let me start with the primary structure of protein. Okay. It is very easy. So amino acids linked with each other in a linear specific sequence. This is the sequence. So serine, threonine, proline, glycine, serine, glycine. So how it is forming the structure? In a linear fashion. Okay, one below the other. They will come and they will form a linear structure with specific sequence. They are called as a primary structure. Okay. So the sequence of this amino acid creates a different protein. If you change the sequence, then they will end up with a different protein. Okay. So this is about the primary structure. You will have a straight line, one after the other. They will come and form the bond and they will form a primary structure. Okay. The next is secondary structure. So in secondary structure, we have two classifications, right? Alpha helix and beta pleated sheet. So let us study about alpha helix first. So alpha helix is a regular folding of backbone of the polypeptide chain. Regular folding of backbone of the polypeptide chain. So this is the polypeptide chain and this is twist into right-handed helix, right? If you see here, it is twisted into a right-handed helix. And how it is existing like this? It is existing like this because of the hydrogen bonding. Because of the hydrogen bonding between the CO group of carboxylic acid and NH group of the amine. Okay, I will show you. You just see here, amino acids are joined together and they form this alpha helix, right? So what is there? Amino acids are there. Amino acid will have C double bond O. So the oxygen of one end is forming the hydrogen bonding with the amine group of the other. And because of this only, they are folding like this and forming the alpha helix. Got it? Now the second is the beta pleated sheet. So what is beta pleated sheet? Polypeptide chain are stretched out and laid side by side. Polypeptide chain are stretched out and laid side by side. I will show you the structure. And they form a pleated sheet like a drapery. Okay, you see here, you can fold this. You can fold this. So this is one sheet and this is the another sheet where we have the CO and NH group. Okay, this is one protein chain and that is another protein chain. So both sheet can form a hydrogen bonding with each other. Okay, these two sheets can held together due to the intermolecular hydrogen bonding. Okay, two sheets can be with each other due to intermolecular hydrogen. What is meant by intermolecular hydrogen bonding? If you have the hydrogen bonding between two molecules, then it is called as intermolecular hydrogen bonding. Okay, so I will show you the hydrogen bonding. So between two sheets, we are having the hydrogen bonding, right? The oxygen of one sheet is forming the hydrogen bonding with the hydrogen of another sheet. So like this, they are held together by this force and exist as a beta pleated sheet in secondary structure. Clear? Shall we go for the tertiary structure? You guys have any doubt in secondary? In alpha helix and beta pleated sheet? Say fast. We have only 5 more minutes. Shall we enter into the tertiary structure of protein, students? All right. Tertiary structure. So it represents further folding of the secondary structure. We have studied about secondary, right? If you further fold the secondary structure like this, you will get a tertiary structure. So this is primary and this is secondary. If you fold this like this, you will get a tertiary structure. Okay. So this gives two major molecular shape. One is fibrous and the other one is globular. So both will fall under this tertiary structure. Okay. But if you talk about the stability, how does this tertiary structure is getting stabilized or how long it can be there and what is the reason for the stability of tertiary structure? To answer all these questions, I would like to give you the force which keeps them together and make it stable. The very first force is the hydrogen bond. Okay, and the second is the disulfide linkage. Disulfide linkage means one sulfur and other sulfur will be linked together and they will act like a hydrogen bond. And the third one is van der Waals. And the fourth force is electrostatic force of attraction. Electrostatic means ionic force. Ionic force of attraction. So these are the four types of forces which makes the tertiary structure more stable. Okay. So you will get question from this in your exam. So it is very important. Okay. The force which is acting on the tertiary structure to make it stable. So these are the four types of force. In case of secondary structure, the force is intermolecular hydrogen bonding. But in case of tertiary, four forces are there. So you should memorize all the four. Okay. This is about tertiary. Now the last is quaternary structure of protein. So in quaternary, we have two or more polypeptide chains. Quaternary structure is composed of two or more polypeptide chain. You just see the structure. It is having a green color, thick kind of folding and the other one is thin, right? So it is composed of two or more than two polypeptide chains or subunits. and we cannot understand the structure. So the name of the structure is complex. Complex, we cannot understand. It is a very complex structure, and it is present in hemoglobin kind of thing. I will show you all the four so that you can understand. Okay. The very first representation of the primary structure is this, and the secondary structure is like this. This is for alpha helix, and for beta pleated sheet also. You can understand with this. And the tertiary structure means you have to further fold the secondary to get the tertiary. And quaternary means you will have two polypeptide chain or more than two. So you can able to see the two polypeptide chain, right? So this is one chain, which is same as that of tertiary structure, and this is one chain, and this is the other. This is the second polypeptide chain. So quaternary is made up of two or more than two polypeptide chain. So this is the representation of all four types of proteins. Okay. Is it clear to all of you? So you just need to know the force of attraction which is acting on each type and the name of those forces. That's it. Shall I go for one last small topic of protein? Okay, I will go to that. The name of the topic is denaturation of protein. What is meant by denaturation? If you increase the temperature or if you increase the pH, not only increasing, you can decrease also. Okay. If you increase or decrease the temperature and if you increase or decrease the pH, the protein will lose its biological activity. There will be one activity for all the biological activity for all the proteins, right? So if you are increasing the temperature or if you are changing the pH, the protein will lose its biological activity. Okay, this is the functional protein, okay, which have its biological activity. If you change the temperature or pH, all the forces which is acting between the polypeptide chain will be broken down. So what will happen at the end? The globules will be unfold. Globules gets unfold and the helix gets uncoiled. They are not together. Okay, they are getting uncoiled and they loses their biological activity. They cannot do the function they have did before. Okay, so you should not change the temperature or you should not change the pH. If you do so, then the protein cannot do its action. Okay. Now the resulting protein is called as denatured protein. The protein which cannot do its biological activity is called as denatured protein. The activity is getting reduced because of the uncoiling of the helix and the unfolding of the globules. Clear? So because of this, if you are changing the temperature, if you are heating it, what will happen? So there will be some hydrogen bonding between them. So that hydrogen bond will be broken down. So the folding is getting unfolded, right? And the coiling is getting uncoiled. And the protein is losing its activity, which is called as denatured protein. And this process is called as denaturation of protein. If you are changing the temperature, it is called as physical change. If you are changing the pH, it is called as chemical change. Okay. If a protein is subjected to physical change, like change in temperature, or if it is subjected to chemical change, like change in pH, the protein will lose its biological activity, which is called as denaturation of protein. Is it clear? This topic is a small topic, but a very important topic. Okay. So there are some examples under each type. You can take those examples like coagulation of egg white will come under the physical change. Okay, curdling of milk will come under chemical change. So you can go through the examples. Coagulation of egg white and curdling of milk. Got it? So this is all about the protein. The next topic is enzymes that we will discuss in the next class. Okay, so far we have completed the carbohydrates part of this lesson and the protein part of this lesson. The next is enzyme. Enzymes, vitamins, and hormones. Those are very, very small topic. We can complete everything in the single class. Okay. Guys, do you have any doubts from today's session? No doubt? Okay, then thank you so much. So take care, all of you. Get ready for your next class. See you all in the next class. Bye-bye.